Hi there everyone. In today's class we're going to look at three main types of GMOs. This is the last part of our three-part series on genetically modified organisms. So in today's class we'll just take a really quick run through of the types of GMOs that are out there, and there are basically classified into three categories. The first one is called transgenic bacteria, transgenic plants, and transgenic animals. The word transgenic basically replaces the meaning of genetically modified. Okay, it just means that it has been altered genetically. So you have a genetically altered bacteria, genetically altered plant, genetically altered animal. Okay. So we'll look at the transgenic bacteria first. It is very common for geneticists to use E. coli, okay, to be the genetically modified bacteria. So genetically modified E. coli, it's basically designed to produce medicines for humans. And one of the medicines that we rely on from this GMO bacteria is insulin. Now, insulin is a hormone that's produced by the human body, and it's used to control the amount of sugar that's in the blood. So that's why we call it blood glucose level. Okay. Now, people who have diabetes, one of the concerns is they might not be producing enough insulin, okay, on their own. So if they're not producing enough insulin, they're not really controlling how much glucose is actually in the blood. Okay, so in order to control that sugar, they have to inject themselves with insulin. But if their bodies can't produce it, where are they getting the insulin from? Well, it's actually manufactured by E. coli. Now, E. coli itself is a naturally occurring bacteria that's found in your large intestines, but it's found in small amounts. In large amounts, E. coli is actually a dangerous bacteria. Okay, most people will probably get food poisoning from eating meat or contaminated food products that have a lot of E. coli. So this one here has been genetically modified with a different purpose. And that purpose is to actually make insulin. But in order to do that, E. coli must have a set of DNA or genetic sequences that instruct the cell how to make insulin. So in order to get that, the DNA is actually used from a mouse or a pig. They're actually not used from humans because we're not really allowed to tamper with human DNA like this, okay, to transfer human DNA from one organism to another. So instead, they use a different mammal. So since mice and pigs also produce insulin just like humans, we use their DNA instead of ours. So the bottom line is this piece of DNA is basically spliced from a mouse or a pig DNA and then inserted into the DNA of a bacteria. And once it's inserted, the bacteria will now only make copies, okay, of this product, of this insulin. Okay, so in a sense, we've turned E. coli into a microscopic factory for producing insulin for human use. Let's look at transgenic plants now. Similar to the bacteria, okay, we do take DNA from another organism. Most of the time it's from another plant, and we then insert it into the DNA of a host plant. The example that I want to talk about with you in this regard is actually canola. Okay, canola plants are pretty common across Canada, okay, especially in Manitoba on the prairies, but not a lot of people know that canola plants are actually GMOs. Okay, the original plant is called the rapeseed plant, and when you look at the original plant and canola plant, they look indistinguishable. Okay, there's no physical difference between the two that you can see, but the difference is actually chemical based. So here's what's happened. The original rapeseed plant was thought to be very acidic. Okay, so the oils from this plant was too acidic to use as a cooking oil. So a Canadian research team operating out of U of M, okay, actually genetically altered the rapeseed plant to be less acidic. And if it's less acidic, it's actually going to be more useful for humans. Okay, so the oil that is produced in canola plants is actually lower in acid content, okay, than the original plant. So that's why we tend to use canola oil as a cooking oil. Okay, it's less acidic and it actually tastes better. Now, the word canola is actually an acronym. Canola stands for Canadian Oil Low Acid. So even the name itself is a reminder of what was genetically altered in order to produce canola oil. Okay. Now, the transgenic animals, there's usually more of an ethical debate, okay, in terms of how we genetically modify animals. For the most part, we either modify them for medicine, okay, or for food, but unfortunately, we also modify them for entertainment. And the first example that I'm going to show you is this little guy. This is a normal zebrafish. Okay, just like any other fish, it's fairly normal. But if you decide to add a gene from a jellyfish to this zebrafish, now you get a whole bunch of glowfish. So the danio here, okay, which is a starfire red danio, okay, this is a zebrafish that has been genetically modified to have a gene from a jellyfish. Okay, since this was successful, they also used tetras and other types of fish and genetically modify them to produce the same kind of coloring effects. Okay, so in this situation, you are using a gene from a totally different organism. Okay, it is an animal, and you're now inserting it or altering it so that you can implant it into a totally different animal. So the glowfish is essentially Cnidarian DNA. Okay, Cnidarian is the classification or group name for the jellyfish, plus a zebrafish. Now, there's no other purpose than entertainment here. Okay, creating a rather stunning looking fish. Okay, but these fish, if you release them in the wild, they will reproduce just like regular zebrafish will. Okay, the only alteration is their coloration. Humans have also tampered with salmon. Okay, but in this case here, they stick closer to home. They basically just use different species of salmon DNA and they just mix them with other species of salmon DNA to produce what we call a super salmon. So it's almost like taking the best qualities of one particular salmon species and another salmon species and making a better salmon species. Using genetics in this way, okay, as a biotechnology can sometimes be useful, but not long ago, we basically can grow beef, okay, in the lab. similar to how we can grow a baby outside the natural womb. Okay, well, to be fair, you're not really growing it outside the womb. You're actually just fertilizing it outside of a human body. Okay, and then you're reinserting the fertilized egg back into a female womb. So, at any rate, we have the technology nowadays to just simply take cells from an organism, okay, in this case, cells from a cow. If we also extract stem cells from their bone marrow, from their tissues, or from their embryos, we can use stem cells to help us turn those cells into muscle fibers. Okay, and the turnaround time is listed here as 6 weeks, okay, in the lab. So it takes about 6 weeks to grow meat, okay, essentially, because that muscle fiber, okay, once it's grown in the lab, they'll basically mix it, mince it, okay, and shape it into traditional hamburger patties. And the person eating them really won't know the difference between this artificial meat and meat that was obtained from slaughtering an actual cow. So in this case here, burgers that are grown in the lab are a little bit more, I suppose, ethical or humane, because you don't have to kill another living organism, but it is made artificially, and some people don't like that. This particular example is technically not a GMO. Why is that? Well, remember, genetically modifying organism means that you're inserting foreign DNA into the DNA of a host organism. Here, you're not altering the DNA. Okay, everything that you're using is from this original cow. So because you're not altering it, you're just artificially growing it. So technically, not a GMO, but it is an example of what we call genetic engineering. Okay. So the last little topic here. At some point, with all of these transgenic bacteria, plants, and animals, someone must eventually come to apply this technology to humans. Okay, so the idea of having designer babies is not a new idea. Okay, it's been around for decades. However, the technology is getting closer and closer and closer to making this a reality. Okay, so we're not there yet, but if you imagine that we were, you can potentially determine what your child looks like before they're even conceived. Okay, by changing, adding, swapping, okay, replacing certain genes in a sperm or egg before those sex cells fertilize. Okay, that's scary sometimes. Okay, now there is potential for good. The pro is, if we have the ability to swap genes in and out, okay, make changes, we can eliminate a lot of genetic disorders, okay, before they even become a problem. We can fix mutations as well before they become a problem. But history has shown that humans tend to use science for terrible things, okay, or at least they can, and when they can, sometimes they do. So the con is, this kind of technology would be very easy to misuse. You might have discrimination and ethical issues. This is actually a topic that I would like to go further with you in class. Okay, so I won't say much more about this now, but I'll plant the idea that what does happen if you can change a person's gene around before they were born? Okay, would you do it? Are there more benefits than what I'm stating here? Are there more issues that we haven't even thought about yet? Okay, so when we get back to class, we will most likely discuss this issue a little bit further. Okay, that's all the time I have for. I hope you enjoyed this GMO series. The next series that we'll look at will deal with cloning. Thank you guys. See you soon.